Welcome to the problem-solving set on genetic libraries. Question 1. Human cDNA libraries are made using mRNA from cells. What, if any, sequence information about a gene is lacking in these libraries? Well, as they say, we're talking about human cDNA libraries. Now, humans are eukaryotes. And the cDNA libraries are made from mRNA, which are reverse transcribed, done by reverse transcriptase, back into DNA, and we call it cDNA because it is copy DNA. Now, this library is just a library of expressed genes, right? But they're asking us what sequence information about a gene is lacking. Now remember that eukaryotes have a different way of producing mRNA, where they go through a maturation period that includes splicing, and that takes the premature mRNA into the mature mRNA that goes out into the cytoplasm. So splicing and the other maturation events take out certain things from the DNA that are made into the mRNA. So first of all, you're missing introns, because those are spliced out so that only the exons are part of the mRNA. You're also missing promoters and enhancers. And those are DNA elements that are part of the DNA that promotes the transcription into RNA, but don't actually make it into the mRNA because they're not part of the gene. And then you're also missing terminators, which again is a DNA sequence that's not actually part of the gene, but it is part of the gene regulatory elements that tells RNA polymerase to stop. So these are the different sequence information elements that you're missing when you just take mRNA and you reverse transcribe it back into cDNA. Problem 2. Consider 4 different types of mouse genetic libraries: a genomic library, a liver cDNA library, a brain cDNA library, and a unique genomic sequence library, where unique means no repetitive DNA. So there's first a few things we have to talk about genetic libraries before we can go ahead and answer these subparts. So remember that a cDNA library is made from mRNAs from the cells. And therefore they are the libraries of only the expressed genes. So we'll say expressed gene libraries. Another thing to consider is that different mRNAs exist in different cell types because of differential gene expression. So a mouse is a eukaryotic multicellular organism, and it has different tissue types, different cell types. So the mRNAs that are expressed in a liver cell can be different from the mRNAs expressed in a brain cell because they're doing 2 different functions. So let's say different mRNAs in different cell types because of differential gene expression. Okay, now we can answer part A. It's saying which library contains the greatest number of clones, assuming inserts of approximately equal size. So this is just something that you need to know, that genetic libraries are stored in clones where the complete genome or whatever piece of DNA is fragmented into smaller pieces and then transformed into these clones, and that's how they keep the whole genome. So it's saying if the inserts, the pieces, fragments of DNA that are put into each clone, are approximately the same size, which library would have the most number of clones, or basically which original genome that you're storing is the largest. So the largest would be the genomic library, right? So it's largest, which therefore would have the greatest number of clones, and it's the largest because this is the entire genome. So all the other three libraries are actually just subsets of this original one, so we'll say this is the entire genome. So even the unique genomic sequence library could possibly be smaller because original genomes, the entire genome, may have repetitive sequences in it, and the unique one removes this repetitive DNA. Okay? Part B says, would you expect any of these libraries not to overlap with the others? Well, even though cell types may be expressing vastly different proteins and genes and making different enzymes, all the cells need to express the same cell maintenance and regulatory proteins. So if you think about it, all cells in a body are going to have to do metabolism and go through glycolysis, so they'll have to be making the enzymes that help glycolysis happen and other cell maintenance functions like that. So you would think that maybe it's possible that the brain and the liver cDNA don't overlap, but in fact they will overlap a little bit because all cells are making the same mRNAs to have these cell maintenance proteins. So we're going to say no, and we'll say that all cells express maintenance regulatory proteins, which means that they must be having the mRNAs for them too, right? So the two cDNAs will definitely overlap at least a little bit, and then these cDNAs will definitely overlap with portions of the genetic libraries because the genetic libraries contain all the genes that an animal has. For C, it says, describe the starting materials for construction of the 4 different libraries. So the genomic libraries come from the DNA taken from the nucleus, right? So we'll say genomic DNA from a nucleus. And then unique is the entire genome minus the repetitive sequences. So we can say it's not exactly sure, you know, if you're not familiar with lab settings, where this unique genome would come from, but we know that it's the entire genome minus repetitive sequences. So this type of genome would be made in the lab and using some technique that can remove these repetitive sequences. Okay, and then the 2 cDNA libraries are made from mRNA from the cytoplasm of the cell that is reverse transcribed back into DNA. And you get this, you can get these mRNAs from the cytoplasm using a poly-U probe. And the U here is for uracil. And that's because, as you might remember, uracil complementary base pairs with adenine, right? So if you have a probe that has multiple U's on it, this can base pair with an mRNA that has multiple A's on it, which, remember, in eukaryotes, mRNAs are polyadenylated, so they have this poly-A tail. And therefore, this probe is a really good way to take the mRNAs out of the solution of the cytoplasm. So these are just, overview of some simple type of genetic libraries, and it's definitely a good idea to know how they differ from each other and also how they can relate and overlap. Problem 3. Sam the scientist created a probe for a polymerase enzyme that hybridized with 3 different cDNA clones. The beginning and ending sequences of the clones were the same, but the middle sequences were all different. How can this be explained? Well, remember that cDNA libraries, or cDNA clones, are made from mRNA, right? That's taken from the cytoplasm of eukaryotic cells. Now, it's important to remember that eukaryotes go through a specific mRNA maturation process known as splicing. Now, splicing is where a premature mRNA transcript is spliced to remove introns, and this can make the mature mRNA molecule. However, with this, it's saying it hybridizes with 3 different cDNA clones, where the beginning and the end sequences of the clones were the same, but the middle were different. So an explanation for this would be alternative splicing. Now, alternative splicing gives more than one specific protein, an mRNA that can encode for that protein, from each primary transcript. So different proteins can be made from each primary transcript. Which alternative splicing can make different mature mRNAs from the same gene. So different mature mRNAs from the same gene. So let's say that we have a DNA gene, and you have the promoter here, and then you have, I'm just going to draw these little sequences as boxes. So you have one sequence here, then I'll draw 3 and I'll label them 1, 2, and 3, and then you have an ending sequence, and then you have the terminator. When RNA polymerase first comes through, you make a primary RNA transcript, which is premature at this point. And this transcript will contain an RNA copy of all of these sequences. So you'll have 5 boxes. But then this premature RNA transcript can go through splicing, and alternative splicing can make different mRNAs, mature mRNAs, from this original transcript. So through alternative splicing, you can get an mRNA that has this beginning sequence that's the same, the end sequence that's the same, and then it has that one sequence in the middle. Another mRNA that has the beginning and end the same, but has the second sequence segment in the middle. Or you can have a third mRNA that has the beginning and the end sequences the same, but has this 3 segment in the middle. So these 3 mRNAs are made from an original transcript, and this probe that the scientists made will hybridize with the 3 separate ones in the beginning and the end, but the middle sequences won't hybridize because they're different. Okay? So this alternate splicing is the answer for this question. Problem 4. Human heart cells are used to prepare 2 gene libraries. Library 1 is prepared from cDNA copied from processed mRNA, and Library 2 is prepared from genomic DNA. For each of the following elements, state whether its concentration will increase or decrease in Library 2 compared to Library 1. So let's write down what our libraries are. So again, Library 1 is the cDNA library made from mRNA from heart cells. And Library 2 is just made from genomic DNA. Now it's asking us, compared to Library 1, whether these elements will increase or decrease in Library 2. However, you want to make sure that you understand what they are asking with concentration. So they're not asking if the number of these elements will increase or decrease in Library 2. It's asking whether their concentration will increase or decrease. Now, concentration is really just a percentage of the whole, right? So let's say you have 5 exons out of 100 parts in Library 1, and then you have 5 exons out of 1,000 parts in Library 2, well, that concentration decreased because now the percentage of the whole is much less. Okay, so that's what they're asking here. So let's look at exons first. So it's saying, compared to library 1, does the concentration of exons increase or decrease in library 2? Well, library 2 is made from genomic DNA, and obviously the genomic DNA has the sequence for exons. However, library 1 is made from mRNA. Now remember that processed mRNA is processed after splicing, where the introns are removed and the exons are basically all that's left in the mRNA, right? So if you think about it, in an mRNA, the concentration of exons are going to be high. But when you add everything else, the introns, the promoters, enhancers, terminators of genomic DNA, that apparent concentration of exons is actually going to decrease. So this first answer is decrease. Now what about 5' untranslated regions? Remember that a mature mRNA has the 5' untranslated regions, then the exons, and then the 3' untranslated regions at the end. So again, because that is a high concentration in mRNA, the concentration in genomic DNA is going to decrease because you're adding in all of those other DNA elements. And remember that, a large portion of actually the human genome, the genomic DNA, doesn't encode for anything. So this concentration is definitely going to decrease when you're looking at it in the genomic DNA. Now what about introns? Remember that we're talking about processed mRNA, which have removed introns. So technically there are none there shouldn't be any in mRNA. But the intron sequences are in genomic DNA. So when you're going from library 1 to library 2, the concentration of introns are going to increase. What about polyadenylated tails? Remember that the poly A tail is added to RNA through the maturation process to mature mRNA. And so therefore, when you're going from mRNA to genomic, this concentration is going to decrease just like exons and the 5' UTRs. And what about promoters? Promoters are DNA elements that are in the DNA, and they signal the beginning of transcription to RNA, but they don't actually make it into the mature RNA. So when you're looking from library 1 to library 2, their concentration is actually going to increase, and that's because you don't see them at all in the cDNA library.